It's my tremendous pleasure to introduce Viola, who's a professor at Dartmouth College. She's just moved there actually from UCSD. And she's going to tell us about her work looking at how salient sounds boost visual-cortical processing and also enhance visual perception. So I'm really looking forward to hearing about this. Viola, go ahead, take it away.
Okay, thank you very much. It's a great pleasure to present some of my work to you all today. Welcome everyone, good morning or good evening, wherever you are in the world. It's a nice morning here in Hanover, New Hampshire. When we look around the world, detailed visual inputs get rapidly projected from the retina to the visual cortex, which then processes that information that leads to our rich visual experience. And traditionally, visual processing is studied in isolation from other sensory systems. However, that research misses one important thing. In the real world, visual objects do not occur in isolation, but they're often paired with other sensory inputs, such as a sound, smell, taste, or tactile information. And so it is important to investigate how visual perception and visual processing more broadly is shaped by inputs from these other modalities. And so today's talk, I want to ask the question how visual processing is influenced by other sensory events, with a particular focus on audio-visual interactions. There are numerous studies that have shown that sounds can influence visual performance. And I'm here focusing just on studies that have looked at spatially specific effects of sounds on vision. And so much research has found, for example, that participants perform better when a salient peripheral sound appears at the same location as a subsequent visual target, for example, in oriented grading, as shown in these examples here, relative to when that sound appears at another location, for example, in the opposite visual hemifield. And so this has been shown in a variety of tasks using different types of behavioral measures. For example, participants respond faster for same location objects relative to opposite location objects. There's also an increase in the detection rate of visual targets when they're co-localized with a sound. And we've also shown that they can enhance perceived contrast of low-level and also high-level stimuli. And they also improve discrimination accuracy. So there's a lot of research already showing that we have these influences on sounds on visual performance in these types of very, very different types of visual tasks. The question that I want to focus on today is what is driving these behavioral effects? So how do salient peripheral sounds influence visual performance? And to do this, in the first part of my talk, I'm going to use human electrophysiology, and I will show that sounds bias visual cortex activity, and that these biases that we see in visual areas contribute to the behavioral effects that we are seeing. In the second part of my talk, I want to dive a little bit deeper into what these biases are, and I will provide some preliminary evidence suggesting that these are primarily due to enhanced processing at the sound's location, with no real signs of suppressive effects. I will talk a little bit about what this means for spatial processing across modalities more broadly, and also for spatial attention. Okay, so let me start by walking you through kind of like a typical cross-modal perception task that we very commonly use in the lab to study these types of effects. So participants in this task perform a visual discrimination. They are asked to indicate whether a briefly presented Gabor patch is oriented clockwise or counterclockwise at the end of each trial. To reduce location uncertainty, that Gabor patch at oriented grading is always briefly masked after it appears. So participants know exactly what happened and they only do the orientation task. We threshold performance using a calibration task at the beginning of the trial. And task just to make sure that participants perform kind of at a rate of 70 to 80% correct. And so critically, prior to the occurrence of these visual targets, we present a peripheral noise burst. So kind of this salient sound either on the left or on the right side. These sounds come from external speakers that we mount to our monitor. So they're really an external space, but they're not critically predictive of the target location or of the target orientation. So these sounds are entirely task irrelevant. And in fact, we usually tell our participants to try to ignore these sounds as good as they can. They're pretty salient, so it's difficult to ignore them, but we really don't want them to kind of engage with the sounds at all. And so when we're looking at performance, we can still split up our trials based on whether those sounds happened to occur at the same location as the visual target, which we would call validly cued trials, or whether they occur at the opposite location of the target, which are invalid trials. And so what you can see here is that we do find a performance difference such that participants perform more accurately when the sound happens to occur at the same location as the visual target, or valid trials relative to invalid trials. And so this is what many other studies, what I just alluded to, have also shown, that these task irrelevant sounds that we think capture attention to the left or right side of space actually influence performance, despite the fact that they're not relevant, that they don't tell participants anything about these visual targets. Okay, and so now we want to see what happens actually, how these sounds are processed and how they affect visual cortical processing. And so you probably noticed in this task that the sounds are always very rapidly followed by these visual targets. So we have a sound and about 100 milliseconds later, or sometimes even less, the visual target appears. And so that makes it actually very tricky to look at the neural activity elicited by the sound alone without being confounded by the visual processing of the visual target. And but we can do a little trick here. So in this task, we can insert trials where the sound appears simply without a target. So in this particular case, actually on half of the trials, we present a sound and then there is no target occurring until the next trial starts, so for about almost a second. And so we intermix these different trial types. So sometimes participants do see the visual target. On other cases, there's no visual target present. This is what we call sound-only trials. And they're all randomly intermixed, so the participants don't know in advance which trial is about to happen. But now we can use these sound-only trials to look at EEG activity elicited by the sound themselves without being contaminated by any visual processing of the visual target. Okay, so that's what I'm going to show you next now. Now I'm going to be plotting the event-related potential over occipital, parietal, scalp sides, and we're going to be time-locking these ERPs to the left and right sounds. So we're going to be splitting them up depending on where the sound was coming from. And in particular, I'm going to collapse here the data across where they were coming from, so left and right side, and also left and right hemisphere, to reel the waveforms that are recorded over the cortex contralateral to the sound, which I'm going to be plotting in red, and ipsilateral to the sound, which I'm going to be plotting in blue. And so here, the sound onset is at 0 milliseconds. Our x-axis shows the time from that onset. And on the y-axis, we have just the amplitude in microvolts, positive plotted downwards here. Okay, and so this is what we find. This is how the ERP waveform looks at. It looks a little funky because we're actually looking at the auditory evoked potentials, but over visual areas, which is not very common. Usually you would maybe look for the auditory N1, but here we're actually looking over visual areas. But the really critical part that we're interested in is, do these waveforms differ depending on where the sound was? And what you can see here very clearly is that around 200-300 milliseconds, the contralateral waveform becomes more positive relative to the ipsilateral waveform. And so this effects last for about 200 milliseconds. It is distributed over parietal occipital lobe. That is very clearly visible when we look at the topographical map of the subtraction of these two waveforms. So we can subtract the ipsilateral waveform from the contralateral waveform. This is what the topography here shows. We're projecting that difference to the right side of the scalp here. And so you can very clearly see that this difference in amplitude, so really what's basically causing the spatial effect, is distributed over parietal and occipital cortex. And so this particular ERP component, so this difference between these two waveforms, we're going to term the auditory contralateral occipital positivity, or in short, the ACOP. Okay, and I'm just going to keep using this term throughout the talk just to make it a little bit easier and to be clear what I'm referring to. Right, this term is really just descriptive. It's just saying basically that it's elicited by an auditory stimulus, but we find this component over contralateral occipital cortex. Now, I just showed you the data for a case where these sounds were completely non-predictive and task irrelevant. And in another task that the same participants performed, we we actually made just a slight change and we actually made those sounds predictive. So on 80% of the trials, they actually were informative to where the target would be occurring. And so in that case, we have longer stimulus onset asynchronies. It takes about a second until the visual target appears. And we really here just wanted to test whether this early activation that we see, starting around 200 milliseconds after the sound, whether that's modulated by a kind of top-down goal, so by maybe endogenous attention or by how informative these sounds are. I'm just going to show you the data for that version real quick here as well. And you can see that we really don't see a difference, right, between the first version where the sounds are non-predictive and this. We can see an overall difference in the shape of the pattern of the waveforms. That might be very interesting, but really these spatial effects, so the difference for contralateral versus ipsilateral waveform, essentially look the same. So what does this tell us so far, the data? We find that the sounds can activate contralateral visual cortex relative to ipsilateral. So we find that these peripheral sounds really induce a spatially specific bias in early visual cortex. These biases start at about 240 milliseconds post-sound onset. They occur regardless of whether the sounds contain some spatial information about the subsequent visual target or not. And possibly one interpretation here is that they're basically prepping visual cortex. They're basically getting visual cortex ready to perform this rather challenging visual discrimination task. Because even though here we're looking at trials where there's no visual target present, participants are nonetheless in a context where most of the time the sound is followed by a visual target. So it might make sense that these two modalities closely interact in this particular task context, because the sounds carry at least temporal predictability, for example, about the visual stimuli. So this really raises the question of how broad this effect is, right? So do sounds only affect vision when the brain is kind of expecting these visual inputs, so when participants are in this cross-modal context? Or is this an effect that extends to other types of tasks and other types of kind of expectancies and contexts? And I think this is an important question because it can really tell us about how sounds, and in particular spatial information about sounds, is processed and about the connectivity of our sensory systems. And so to test this, in another experiment, we changed the context rather radically, and instead of participants doing a visual discrimination task, we actually asked them to do an auditory, difficult auditory discrimination task. So exchanging our kind of lateralized visual target stimuli with lateralized tone stimuli. So participants are doing a pitch discrimination task in this case, but these auditory target tones, they're still preceded with these peripheral noise bursts, okay, on the left and the right side, exactly like in the first experiment that I showed you. And so now we can look at the activity elicited by these left and right peripheral noise bursts that are task irrelevant, that participants are not responding to at all. But again, participants are now in this auditory context. So they're listening to these left and right tones that subsequently appear here much later, around 600 milliseconds later. And so I'm going to show you exactly the same plot that I showed you before. Okay, so again, recording ERPs with the onset time locked to the left and right sounds, and dividing them up contralateral or ipsilateral to the sound. And so here we find exactly, again, our ACOP component. Okay, so again, we find that these peripheral noise bursts elicit a kind of biased visual cortical activity in a spatially specific way, even in this task where participants never see a visual stimulus. They do have their eyes open, they're staring at fixation cross, but they never do a visual task. They never see a visual peripheral stimulus. So all they're doing is hearing different sounds. And nonetheless, these peripheral noise bursts activate and bias visual cortex around the same time as we've seen before. However, in both of these tasks, the targets, in one case the visual targets, in the other case the auditory targets, still appear at a predictable time. And the task itself is somewhat spatial because the targets themselves are actually presented at the left or the right side of space. And so kind of to eliminate those two things, to also take away the predictability in time and also the spatial part of the task, we ran yet another version, in which case basically we're just presenting a random stream of auditory stimuli. Okay, so there are either these left and right bursts of noise, and they're also about half of the time there's a central target tone. And all participants are doing is listening out to the central target tone that's very easily discriminated from these peripheral noise bursts, and all they do is press a button whenever they hear that target. And so here the idea is really that the task is non-spatial anymore. It's a pretty trivial task for participants. And you can also see that we're jittering the timing between all of these stimuli. So really it's just a random stream of different sounds that participants are listening to. And the question here is, do these peripheral noise bursts again elicit and kind of bias visual cortical activity? And what we find is that yes, they do. So once again, around the same timing, we find this ACOP. So we find that these peripheral noise bursts elicit a larger amplitude signal over contralateral relative to ipsilateral cortex. So now I've shown you a bunch of experiments where we find that the salient sound can boost contralateral relative to ipsilateral visual activity in cross-modal versions of this task. So when participants actually performing a visual discrimination task, in purely auditory task, but also in this, what I'm not going to even call really task, but just in this like kind of random stream of sounds where participants just once in a while press a button whenever they hear a target tone. And so across many different experiments, now I just showed you some of them, but across many, many different experiments in my lab, we have found that a salient sound can boost visual cortex activity. And it seems to be the case that this effect is very robust across different task contexts, really suggesting a tight link between audition and vision that cannot be overcome very easily, at least within a single experimental session. And so these interactions, I think, suggest that they're either very well learned throughout life or even possibly hardwired. And one question these data raise is what functional pathways that are involved that lead to this effect. We know this is pretty tricky to get from EEG, but we do know that there are direct connections between auditory and visual cortex based on other literature. But I think the rather late timing of this ACOP, so around 240 milliseconds on average, suggests that not these direct pathways underlie the effect, but rather indirect pathways are involved. And so possibly it could be that these salient sounds essentially act kind of as an exogenous cue that activates the attention network, which then sends kind of these attention hotspots back to visual cortex. And this interpretation is also supported by the spatial specificity of the effect, right? So the brain region that is responsible for the sound location gets activated relative to the unattended or opposite location. And so I think this spatial selectivity is really the most exciting part of this data and has really important implications about spatial processing across modalities. It suggests that there possibly exists a sound-based spatial topography coded in visual coordinates, possibly because of the spatial dominance of visual cortex. And such a mapping seems to exist at a minimum at the hemifield level, so left versus right, which is what we're looking at here. And it may well be the case that that's all there is. It's just this kind of hemifield mapping, and possibly also only kind of in the periphery where vision is particularly poor, detrimental. But of course, another possibility is that there is actually richer spatial information about sounds that is projected back to visual cortex, possibly across the entire visual field. These are all, I think, important and interesting questions for future research. But what I want to focus on today is the link to selective attention. And in particular, the question whether these biases that we're seeing are due to enhanced processing at the sound's location or kind of suppressed processing at the other location. And so many theories of spatial attention, prominent theories of spatial attention, have proposed that attention operates via excitation at the same time via inhibition, based on the idea that visual processing is a strictly limited resource. And so if processing gets enhanced somewhere at one particular location, it basically consequently necessarily has to be decreased elsewhere. And there's a lot of evidence that kind of in top-down attention, so when we tell participants what and where to attend to, that we find kind of these two flip sides, excitation and inhibition. But very little is known in the context of these cross-modal tasks, and also in the context of kind of exogenous attentional orienting more broadly. So if we think about these salient sounds as kind of an bottom-up salient cue that orients our spatial attention reflexively to the left and the right side, that such that captures our attention, that could be kind of thought as a cross-modal exogenous attention. And so I think it's important to explore this question, whether these effects are really due to excitation or inhibition. And so this is what I'm going to focus on now in the second part. And I'm going to do this in three different ways. We're going to look not only at ERPs, but now also at alpha oscillations, which are a very prominent marker of studying suppressive effects in particular. I'm going to try to link these different neural measures to behavior and see if we can kind of learn from the pattern what type of behavior they predict and whether they can tell us something about excitation or inhibition. And then lastly, I'm going to show you some very recent experiments that we ran trying to isolate neural enhancement and suppression using experimentally. So occipital alpha oscillations are sensitive to visual processing. It's a very prominent, well-known marker, and it's very well known that this 10 hertz oscillation around 10 hertz is associated with kind of the amount of visual processing, very broadly speaking. So decreases in alpha power have been associated with more visual processing, and increases in alpha power have been associated with less visual processing. And there's kind of prominent theories that really suggest that these alpha changes, in particular increases in alpha, reflect kind of top-down control of suppressive mechanisms in particular. And so in the task that I just showed you, really in all of the tasks that I just showed you, we can not only look at the event-related potential, what I've done so far, but we can also look at alpha activity and see if we find changes in the occipital alpha rhythm that have been very strongly associated with these kind of biases in visual-cortical processing. And so I'm just going to show you here one example of this last task that I showed you, where we just present these kind of random stream of sounds. And we're going to again be time locking now our EEG activity to these task equivalent noise bursts, depending on the left and the right side. But instead of looking at the ERPs, I'm going to be now showing you time frequency plots. So here on the x-axis, again, time, 0 is the onset of the sound. And then on the y-axis, you now see not just amplitude, but you see the frequency. Okay, so from kind of 0 up to 30 hertz, what I'm plotting here. And the different colors denote how much activation there is. So blue is a decrease in alpha power, and red would denote an increase in alpha power. So here I'm now showing you the time frequency plot for the activity recorded over the hemisphere ipsilateral to the sound. And you can see that we kind of see a small decrease in alpha power around 10 hertz. Now I'm showing you the activity contralateral to the sound, where you can also see kind of this clear blue line around 10 hertz of activity. It's a little bit easier to see if we do the difference between these two time frequency plots, which then very clearly shows that we have a stronger decrease in alpha activity over the hemisphere contralateral to the sound relative to ipsilateral. And you can see here that this decrease in activity really happens around the same time window that we saw for the ACOP, so around 200 to maybe 500 milliseconds. Of course, there's some temporal smearing here, but it really closely actually resembles the time course of this ACOP component that we measured. And so I just want to point out a few things here about this data, right? So we see that alpha activity contralateral to the sound decreases relatively quickly and transiently. And relatively quickly and transiently, I'm here thinking about a ton of work on endogenous spatial attention that also finds these lateralized biases, so when participants are told to attend to the left or the right side, but usually they occur much later in time, around 500, 600 milliseconds is maybe when these alpha changes are beginning, but here they're relatively fast, really suggesting that alpha is not necessarily a sluggish response, but can actually be triggered and elicited rapidly by salient peripheral stimuli. And these alpha changes, again, just to remind you all, are elicited by task-irrelevant peripheral sounds, right? So we're looking at activity here, the occipital alpha rhythm, so over visual cortex, but these changes are triggered by sounds that are presented about 100 milliseconds beforehand. And so they're not due to kind of this top-down control suppressive mechanism that often alpha changes are related to. And one other point that I just want to kind of allude here point people to is that what we see here is that these alpha changes, decreases are bilateral. So we see decreases ipsilateral and contralateral with a stronger decrease over contralateral visual cortex. And so this is actually very different from many, many reports and kind of top-down endogenous attention that very often report changes that are due to ipsilateral increases in alpha and or contralateral decreases. But what we see here is really just that overall kind of alpha is decreased, just biased to be more decreased contralateral to the sound. And I think that could be potentially suggesting that there is kind of a difference in how these alpha modulations occur between different types of tasks. Okay, so overall, the fact that we do find these decreases are consistent with the interpretation that what we're seeing here is a visual enhancement, right? If we think about alpha decreases really kind of reflecting in increases in visual processing, not suppression of visual processing, then this pattern of data is consistent with the idea that the sounds increase or enhance cortical processing in visual areas. Now let me move to the kind of second point here. So in another task that's really similar to the ones that I showed you, we try to link the ACOP and also alpha activity trial-by-trial performance. And so here I'm showing you the task that we're using. Again, it's really similar to the task that I've showed you before. In this case, it's not a Gabor patch that participants are discriminating, but it's an L versus T task. But really the critical difference that you have to pay attention to is here that we expanded the time a little bit between the sound and the visual target. So here we have 300 milliseconds between the sound and the target, much longer than what we usually use. Usually it's about 60 milliseconds, much more rapid than that. But if we only have 60 milliseconds, that does not allow us to link trial-by-trial variation in performance to trial-by-trial variation in the ACOP or in alpha activity. But here we have just enough time basically to look at neural activity and behavior within the same trial. And so this allows us now to split up the trials based on whether they were validly cued or invalidly cued and whether participants responded correctly or incorrectly, and kind of look at how magnitude of ACOP or alpha activity relates to behavior. And so let me walk you through the logic here. The prediction here is that if the ACOP reflects primarily enhancement, you would expect to find a larger ACOP, so more positive activity in the hemisphere contralateral to the Q relative to ipsilateral, preceding correct than incorrect discriminations for validly cued targets only, but not necessarily any difference for invalidly cued targets. Conversely, if the ACOP reflects primarily suppression, you would expect to find a larger ACOP, so more positive again in the hemisphere contralateral to the Q, preceding incorrect than correct discriminations of invalidly cued targets. Okay, so really what this means is that we basically want to kind of figure out here what the ACOP predicts, right? Does it predict performance on valid trials or invalid trials? And from that, we can make some inferences about where the action happens, right? So whether it facilitates processing at the validly cued location or kind of hurts performance at the invalid cued location. And obviously, you could also imagine a mixture of these two, right? I'm just kind of putting up here the two most clear results. And so let me show you what we find in this paper. We find data consistent with the excitatory or enhancement account, such that the magnitude of the ACOP, here plotted on the y-axis, predicts performance only on validly cued trials with no difference on invalidly cued trials. And so this suggests that the ACOP enables more effective visual discrimination at the cued location and implies that the ACOP does not necessarily reflect suppression at the uncued location. And so interestingly, we find exactly the same pattern of data for our alpha amplitude. So here alpha and ACOP really seem to kind of go together. We don't always find that in all of our experiments, but here it really seems to be the case that lateralized alpha predicts performance again only for valid trials, possibly suggesting that these alpha changes reflect facilitation at the attended location with no suppression at the unattended location. And so I think this is, while this is rather indirect evidence, it at least points to the fact that possibly these two neural markers are really related to kind of enhancement of neural processing at the location of the sound. Okay, so now lastly, I want to provide one more piece of evidence where we try to experimentally isolate this neural enhancement and suppression, an experiment that was just recently finished by my graduate student, Jonathan Keefe. And so in this task, we're going back to our lovely Gabor patches here, but the same task essentially that I showed you at the very, very beginning of the talk. So participants performing a discrimination on these oriented gradings. We're presenting our kind of standard left and right sounds, but now we're also adding a central sound. And so the central sound is basically supposed to serve kind of as a baseline here. Really, the idea is that the sound is matched in all low-level features and also has some the same kind of temporal predictability that these left and right sounds have. But the only difference is that it does not cause any spatial orienting response, right? So the sound comes from the center. It's very clearly kind of, you don't orient left or rightward. It should not cause any lateralized changes in visual cortex, but it might still cause changes just due, and many studies have shown just even central sounds activate or can elicit activity in visual cortex. And so really want to get kind of at this general excitatory or whatever response that these central sounds have, but then compare it to these spatially peripheral sounds where the orienting response happens. Just like in all other tasks, the sounds are not predictive at all of where the visual targets are about to appear or when they're appearing. We actually have here different types of SOAs. So either the visual target appears right after the visual cue, the auditory sound, sorry, and that allows us to look again at behavior as usual, but then we also have trials where there is a rather long delay, around 600 milliseconds between the sound and the visual target. And we also, again, have trials where there's no target at all. And so these latter trials, the long SOA and target, we can use to look at the EEG activity without the contamination of the visual target. And so that's what I'm showing you here. So you're fairly familiar now with these waveforms. We're again time locking to these peripheral noise bursts. And here what I'm just showing you is again the waveforms to the left and the right sounds. So we find again here in this highlighted region the ACOP, similar distribution as I've shown you before when we look at the topographical map. So really the main question is now, where does the central sound fall, right? Where does the waveform of the central sound fall? So if the contralateral waveform differs from the waveform elicited by the central sound, for example, it is more positive than the central sound one, this would be consistent with the contralateral cortex getting enhanced relative to that baseline. And likewise, if the ipsilateral waveform differed from the central tone or central sound, for example, it's more negative, relatively speaking, this would suggest a role of suppression, so less activity ipsilateral relative to this baseline, to the central sound. And so what we find is that really the waveforms to the central sound kind of lies on top of the waveform to the ipsilateral sound. It's even a little bit hard to see. Here I'm plotting, to make it a little easier, the mean amplitude for our three conditions, so contralateral in red on the left, followed by ipsilateral, and for the central tone. And so this really suggests that kind of all the action happens, all the difference happens in the hemisphere contralateral to where the sound was, with no additional kind of costs ipsilateral. And so this is broadly, again, consistent with this idea that the sounds primarily enhances visual processing at its location, but does not have any kind of costs or suppressive or inhibitory effects in the opposite hemisphere. And so this pattern of neural data that we find here very nicely matches our behavioral data in this task, which I'm now plotting here. So again, this is just proportion correct for our three trial types, valid, invalid, and central. And we can also see that when we're just kind of looking at the cost benefits of these sounds on visual performances, what we see is that there's really just benefits for the validly cued trials relative to invalidly, and then our baseline, the central sound. So consistent with our interpretation of the neural data, that really the sounds are primarily facilitating visual processing and not hurting visual processing at the unintended location. And so kind of looking at these different, right, so in the last part now I showed you that this like stronger activity contralateral relative to ipsilateral, and the central sound is also consistent with enhancement. So I think we kind of are getting pieces of evidence looking at the data in different ways and looking at different types of experiments, suggesting that these biases that we see in visual cortical areas are primarily driven by enhancement, neural enhancement in visual areas. So to sum up, I started with showing you that sounds influence visual perception, in particular by increasing visual discrimination accuracy. That's the main task that I talked about today. And then I showed you a series of studies where we found that salient sounds very robustly bias visual cortical activity, and that these biases even occur when there's no visual targets presented ever. So when participants are in completely auditory contexts. And the kind of last part of my talk, I showed you some evidence suggesting that the sound-induced effects on visual performance are largely driven by excitation or enhanced neural processing at the sound's location, with no effect really at the unattended location. And I think this might have important implications when we kind of more broadly think about spatial processing across modalities, but also if we kind of compare effects of exogenous attention, which I think is basically what we're studying here in a cross-modal context, and endogenous attention. In endogenous attention, many studies have shown, and there's still currently a lot of ongoing work showing that endogenous attention enhances and at the same time suppresses information processing at unattended location. So there might be a really interesting difference here of how these two types of attention operate in enhancing visual perception. Okay, so I want to thank the wonderful people that helped with all this work. A particular shout out to Jonathan Keefe, my graduate students, who's been working a lot on these projects, and also all the other co-authors of the work that I presented today. I want to thank my lab at UCSD and at Dartmouth, and I want to thank you all for listening.
Great, thank you very much. Wish that the entire audience could clap, of course, but this is...
Thanks to you for clapping.
Yes, that's fascinating. The first question that I have, and then we can get to some questions from the audience as well. So are people hearing this, and maybe you mentioned this, through speakers in front of them or through headphones?
Yeah, I think I mentioned this very, very briefly. So it's all through speakers that we mount to the sides of the monitor. We have not done this with headphones. I think my intuition is that it's actually important that these speakers are kind of an external space, so that you really kind of get this. I think it's very important that you kind of get this like external space perception. Of course, you can titrate it with headphones. So I think it could work with headphones, but we actually haven't tried it.
Yeah, how selective do you think the spatial location is? Like, in other words, how important is it that like the sound source is directly there versus if you had like a speaker like perpendicular?
So we know from many pilot experiments in the lab, and just kind of like trying things out differently, that it's actually really important that the visual target location overlaps very precisely with the sound location. I think that might vary by where you are in the visual field. So in all of those cases, and maybe I didn't emphasize that enough, all these sounds and the visual stimuli are really far out in the periphery. So we're talking about like a visual angle of like a minimum of 24 degrees to get these really clear and reliable spatial effects. So when you move closer, the behavioral effects, at least we know, kind of attenuate. And so there could be something about really this like peripheral part of the visual system that I think interacts maybe with like the auditory system basically particularly well, possibly because visual perception is kind of more poor, right, in the periphery. But in these like peripheral locations, it's really important that we have very close mapping. So we're actually adjusting the sounds. We have the speaker kind of next to the monitor, but we're actually adjusting the sound to be perceived exactly to where the target is by adjusting the volume between the two speakers. And that seems to be important to kind of get the most reliable behavioral effects, at least. So there seems to be a pretty close mapping, maybe surprisingly close.
Have you tried it with using, like, for example, the ILD instead of like a speaker?
No, we've only done it with speakers. We haven't tried any of the other ones. And I think, I think it could work if you, I don't know. I'm really not sure, actually. I'm always skeptical, so that's why we never tried it, I guess.
Yeah. Another question about the potentials. I noticed the very first EEG you showed, the ERPs rather, there was not a large positivity associated with the auditory stimulus, whereas in the later ones, there was this very sort of clear positivity at around 200 milliseconds. And I was wondering if you think that that is somewhat equivalent to like a visual P3, but occurring earlier. I know that everything in the auditory domain is kind of shifted a little bit earlier.
Oh, I see. So early, you mean like early in time in the ERP waveform, right?
Yeah.
Yeah. So, I mean, we see when we look over auditory cortex, we see the lateralized effect, a small, much smaller one, but we see a lateralized effect as you would expect in the auditory N1, right, around 100 milliseconds. So when we look over auditory cortex, we see a small contralateral increase in the auditory N1 component, and then really nothing happening afterwards. So then it's completely just over occipital scalp areas. Right. I don't think this is a visual P3 because it's not related to a visual.
Right. So I was thinking like the sort of analog of.
That's the right way. I mean, we think it's like a new component that we found, basically, and it's not really related to like the visual P3, but it's true that it's rather late, but it's not triggered by visual stimulus, it's triggered by a sound stimulus. And so it's a it also, like, the pattern looks pretty different. It's kind of longer lasting, right? It's really like 2 to sometimes 300 milliseconds long. So it's almost slower than any type of P3 component.
Oh, sure. Yeah, yeah. So the ACOP, right? That's the sort of
Yeah.
bilateral difference. I was thinking of it looks like there's also a not difference wave, but like a bilateral,
Oh,
that big huge peak, right, which is bilateral.
got
And
it.
that kind of might be the equivalent of sort of the P3 to a visual event.
Maybe the P3, I think we've been thinking about more because we see this really interesting difference that I just very briefly, I think, pointed out between when the sounds actually carry predictive value versus they don't carry predictive value. And we see that the overall waveform shifts when they carry predictive value about the location, that it becomes more negative overall. So we have this kind of like upward shift of the entire waveforms. And I think that might be related to kind of like sustaining attention maybe at that location, right? Because they actually, participants are trying to use the sounds. And so it could be, I don't know if it's like the P3 or maybe more something like a sustained activation, negativity that kind of tags that particular location and kind of sustains there over time, and is really related to more these attentional, kind of endogenous attentional processing. That makes sense. But I think we would have to do more manipulations like that to actually kind of get at the overall shape of the waveform, which we just haven't done a lot. We really just have this one comparison where kind of popped out to us that the predictability of the cue actually shifts these waveforms around.
Yep. Right. Well, let's dig into some of the questions here. The first one is, could the ACOP be a general spatial expectancy signal in the parietal cortex, I guess, relative to something specific to the auditory?
Yes, and I think that's actually likely the case. I would add that we don't think it's just the parietal cortex. We really think, actually, in the really early studies that we did, we always found mostly occipital activity. We also did like some distributed source modeling and dipole modeling. Of course, these are all just models, but it always really fell into visual cortex in particular. But we often kind of see this additional parietal component. So it might be a mixture of both parietal and visual, I think, components that are inside the ACOP. And we actually ran a study just looking at visual cues, and we find essentially the visual counterpart to the ACOP, which we call the VCOP. And it looks really strikingly similar. It's much more difficult to interpret because, of course, when you look at activity elicited by the visual peripheral cue, you're going to get early difference in the waveforms just triggered by the visual response itself, right? So you have big difference in the P1 component, N1 component, but all of that is followed by a kind of slow wave contralateral larger than ipsilateral, exactly resembling kind of the results of the ACOP. And we have a recent paper kind of comparing these two components, suggesting that this is a general kind of marker of exogenous orienting. So I think that's true. I think it's just much, much harder to interpret in the visual domain, basically.
Yeah, it's nice that you have the auditory stimulus, makes it a lot easier.
Yeah, that's right. That's one big disadvantage of auditory stimuli.
Yep, that's cool. Next question from in the Q&A is by Jan Hartung, and he asks, he says, cool talk, thanks a lot. So you said that the sound might prepare visual cortex because in this specific context, the sound is associated with a visual stimulus most of the time. If you decrease the proportion of trials including a sound, does the effect on visual cortex disappear at some point? So I guess like in the extreme, you might just have like one trial. Like, do you think it would work in that case, I guess?
So if we just decrease the proportion of sounds overall, is that the question?
Right. So the sounds were very rare.
So I actually think it would probably increase the ACOP, because I do think it has a lot to do with just kind of how salient these sounds are. We try to make them so salient that they basically stay salient throughout the whole experiment. Even though people have been in there for an hour, they still kind of capture attention and are salient enough to trigger the ACOP. Mm-hmm. My suspicion would be if we made them even more rare, that actually the ACOP would increase because they kind of engage, they like basically capture attention more, right? Because I really think it's an attentional effect. And we have not done, of course, we have the data to actually look at kind of like trial by trial variation of the ACOP to see if potentially, if we happen to have 3 left sounds in a row, does the third one actually produce a smaller or bigger ACOP, right? To kind of look at these sequential effects. Jonathan is actually currently working on that, but we don't know the answer to it yet. We haven't really done it systematically. And so we're kind of looking at the data that we currently have to see if we find effects that I think could get at that question. But anyways, I think my intuition would be the other way around. I think actually more rare sounds are going to produce a larger ACOP because they engage kind of attention more, essentially.
Yeah, no, I completely agree with that. I think that they would. it just sort of pop out.
Yeah.
Someone asks directly in the chat. Someone was wondering if the same component would be there without any audiovisual integration. And I think that you showed that. Is that true?
Yeah, I think yes, yeah, because we have these tasks where participants do never see a visual target. And so we even then see the ACOP, right? So if they just hear this random stream of sound, I think that's kind of the strongest experiment that we have, showing that where these sounds are just all in random order, participants never see visual stimulus, and we get basically the same sized ACOP that we find in the other experiments. So I think this is basically suggesting that there's a direct kind of interactions in spatial processing between audition, the auditory system, and the visual system. We don't need the visual target, right? So it really gets, that's probably what the question is about, like, do the broad multisensory integration question, do we have cross-modal interactions even without kind of the second modality being present? And I think we do have evidence that that's the case, that the visual target or visual stimulus does not need to be present for these interactions to show.
Right. Oh, we have a question from YouTube. Aparna Pandey asks, do you think that the facilitation on visual stimuli would be possible for more meaningful auditory sounds or related sounds? So like, for example, a case where the auditory sound is related in some way to the visual stimulus?
Yeah. So, I mean, there's a bunch of work kind of on using different, how like features, right? So for example, like high pitch sounds are associated with, I don't know, brighter stimuli. So maybe the question is getting a little bit like, that's not really meaningful, but there's a bunch of research kind of showing that there's like these correspondences where also behavioral performance is facilitated. Yeah. And one of my grad students, Jenna, she's really interested in these correspondences, and she also re-replicated these behavioral effects. I think that people are finding, but I think those are fundamentally different mechanisms. I really think this here, these spatial connections, I think are really robust, possibly even hardwired. And I think as soon as you kind of move into maybe looking at feature correspondences, they're probably happening at, I think, like at a higher level, essentially, right? And I mean, we are doing a little bit of this work, so looking at different features. And another grad student of mine is working on really high-level stimuli, so really looking at how kind of auditory, the sound of an auditory scene, for example, right? Like listening to a football stadium or something when you walk in a football stadium, how that affects the visual discrimination of real-world objects. And behaviorally, we find associations between all of them, but I really think that that happens at a like completely different level, right? Those are like semantic associations probably that are really well learned. And so I don't know exactly, I think at this point, how to compare them. I do think those associations definitely exist, but I think here we're looking at something like really low level of kind of looking at like spatial maps across sensory modalities, which I think is just a more possibly hardwired mechanism. I don't know if
that makes a lot of sense.
Yeah.
I was wondering also if you've tested how specifically in time the relationship holds, right? So normally, like with exogenous cueing, you see a sort of very brief window in which it's effective on the visual domain, right? How does SOA affect here?
So we've the ACOP component and also the alpha modulations, they really basically always start exactly at the same time. I mean, more or less around 200 to 240 milliseconds. They do, so when we do manipulations such that the sound, for example, becomes predictive of the target location, they basically can last longer. Actually, the ACOP not at all, but alpha. So alpha activity seems to be modulated by kind of the top-down goals in addition to this reflexive oriented response, whereas the ACOP really seems to be primarily driven by just kind of this exogenous part. Mm-hmm. And also behavioral, we find actually really similar types of effects in terms of timing as you would for visual cues. So it's, I think, this kind of exogenous attention is probably like very broad across these modalities. Mm-hmm. Something that I didn't point out is, but the ACOP time window corresponds really, really nicely to this time interval of kind of broad exogenous cueing effects, right? That we usually kind of say peak at around 100 milliseconds or last up to 150 milliseconds. And so the ACOP, it's a little bit complicated because you actually have to like subtract how long it takes to reach visual cortex for a target stimulus, right? Yeah. But it really falls if you present the sound and 100 milliseconds later you present a visual stimulus. Basically, it takes another 100 milliseconds or 80 or so.
Yeah,
so that visual stimulus is actually in visual cortex, and that's exactly when the ACOP starts. And then it lasts around 200 milliseconds. So it's really in that time window that many, many visual exogenous studies have shown where the peak is of exogenous attention. And so that's also right why this link, I think, to spatial attention and exogenous attention more broadly makes a lot of sense.
Right. So we've only got like perhaps another minute or so, and we're going to cut off probably pretty abruptly. But let's ask one more question here, which is whether you think that these findings translate in some way to blind or visually impaired people.
Oh yeah, we actually have a study on that that was conducted by Mori's lab at the IIT in Italy. And so they tested blind subjects, and basically it's similar paradigms that I showed you today. And so interestingly, in congenitally blind people, so those that never have no visual experience whatsoever, we find that the ACOP exists. If anything, it's bigger than in non-blind people. Really interesting finding, really suggesting that there's some really like hardwired, interesting relationship, I think, between auditory processing and the visual cortex that doesn't need the visual experience for that wiring to occur. And so yeah, we have those results. They're published. People can look it up. Monica Gori is the last author. Yeah, I think it's really interesting and really suggests that there's a kind of very robust wiring between these two systems in terms of spatial processing.
Is that Gori as in Gorey?
Yes.
All right. Okay. Well, thanks again.
Thank you.
It was excellent.
For having me.
Thanks also to the audience.
Yeah.
All
Thanks
right.
everyone for listening. Have a good
everyone.